I prepared a presentation without knowing more or less the context, but my idea is to show you a little bit of the biology and the fascination that is behind or below our feet, and then to introduce you into the way how we can use this as a tool to produce better and in a different way. And our goal is to show that in order to achieve these aims, we need to transfer this knowledge and to share it with the society. But this can be done not only in a specific way, that is, we working as a source of knowledge that most of the time we are not, but with the interaction and involving all the citizens in the process. And in this regard, I will finish telling you a few words about the ECO project, that is a soil project focused on citizen science. So, having said that, and having to confess that I'm very jealous that I'm not there with you, because I heard that the weather is wonderful and everything is very pleased. I would like to share my presentation with you. Usually I have everything in English, but today I made an effort to have it in Portuguese. Sorry for that, but I will talk in English. So let's start by assessing. And please, if you have questions and if you want to share some ideas, some doubts, to add something, please be free to do it while I'm talking, because I think that enriches the communication. So my first slide aims at showing you how soil, what is the composition of soil. And all of us have the idea that soil is mainly formed of minerals. Then there is a component that is full of air or water, depending on the weather conditions. And there is a small fraction of organic matter. This organic matter is really the key process in the soil functions. And most of it is that, apparently, at least it's that, is remains of living bodies. But then there is this green piece here that is formed by organisms, living organisms. And these are bacteria, earthworms, animals, microfauna, algae, fungi. Fungi are very important components of this system. And of course, this represents a very, very high biodiversity. And when we have a soil, or when we think in the soil system as a whole, we can imagine that most of the species that exist are living in the soil. From all the species that are known, 59% of them live at least one part of their lifetime in the soil. And this is true for many, many types of organisms. For instance, if we see these ones here, the Enchitraida, 99% of them live in the soil. Bacteria is more than 43% of the bacteria are present in the soil. Nematodes also, and then there are others like Arthropoda, Archaea, and Mammalia that are more abundant in all the other components of the ecosystems, but there are always some that are present and that characterize the soil. So this slide provides us the relevance of soil as a hotspot of biodiversity. But if we want really to understand it, and I'm sure that you have your boots full of soils, your hands, that you have been holding soil in your hands during this morning. So in a handful of soil, we may have more than 500 species of insects. This is a fantastic number, and we have to understand in which form they are present. For instance, protozoa, we may have between 100 and 500 species. If we go to bacteria, this is billions of bacteria. And look at this number, 50 kilometers of fungi. This is a lot. 50 kilometers is from Lagos to Portimão or more. So this is a huge amount of a huge length. So what I want for you to think about is how full of life soil is, but how diverse this life is. And even I think we have the feeling when we look at these numbers. I would like you to have ideas and understand it. So I presume that there is at least one farmer in the room. So can I have a farmer that one? Can we identify a farmer or somebody that is involved in farming?
Sure, at a high level, Walt is involved.
Okay, so
A couple of others.
you are very familiar with plants. So my question is, you can see the leaves here, and I'm not going to ask you how many leaves a plant has, but I'm going to ask how many apices, root apices are in a plant.
You mean the root hairs?
Not the root hairs. That is a much bigger number. It's each root, each secondary root has an end. This end has a meristem, has a growing point, a growing tip.
Yes.
So how many of these growing tips are present in a maize plant or a carob tree or...
I'm not the one to answer, but quite a few more than the leaves, that's for sure. I don't know, maybe.
Good answer, but I want a better one.
Yeah. Let's say 1,000.
1,000. That's a good start. More.
More, okay. 10 times.
Much more, much more. It's it's in the order of 10 millions. So this, what I want.
Okay, go ahead.
Sorry, please.
I was wondering what sort of plant. You said corn?
Yes.
Wow.
Yes, corn. But it can also be a carob tree or olive tree.
Yeah.
This is always in the order of millions. It can vary between 10 millions, but it's always in that order of magnitude. And what I want you to see, to have the image, is that we are talking about a small item in the soil, but there are a lot of them, and this makes the difference. This changes the soil, this changes the environment, this changes the chemistry and the functionality of the soil. And the same happens with the living bodies that are there. So what is this?
Like a mold.
A mole, exactly. Most of the farmers do not like them, but they can be very relevant because they make holes, they aerate the soil, they remove it. It's they have several uh, functions that are very important, and they also, from the biological point of view, they are very well adapted to live in the soil, a place where there is no light, okay, and this is determinant, but where the communication between organisms is a constant and is a determinant for a good soil. So this one you also know about. You have a picture, we have a name. They are insects. They are those that sing a lot when it's not really singing, but it's they control their body temperature through the movement of their wings, and they make funny noises in the summer, especially when it's quite hot. And they change their exoskeletons, and after that, they can spend a long time. It's a time that goes up to 17 years in the soil. So this is to for us to understand that when we look at the soil functions and we see pollinators and pollination as one of the main soil functions, it is related with these life cycles. Many insects have life cycles where they stay in the soil for quite a long time. And if the soil is not in good conditions, their cycles are disrupted and their lives are endangered. This is a beetle, and everybody knows about this one. All of us play with these balls and then are very upset when we discover what they are made of. But from the ecological point of view, these are balls of life. They are living balls where seeds can find the right conditions to germinate and to develop. And this, what is this?
Worms.
A worm, a worm. So, and if I say that I do not see a worm here, that I see an incubator, would you agree with me? Also, of course. Why? Because when we have this type of animals, do you know how many herds the her earthworm has?
Five is the answer.
It's about 70, no? So there are so many of these strange things happening in the soil. These are animals that are very difficult to kill because you divide them in two, and instead of a dead animal, you have two living animals. It's not always like this, but most of the time. And why are they incubators? They are incubators because they eat the soil and then they excrete the soil after incubating it with a very peculiar community of bacteria. And these are nitrogen fixers, is methane producers, and therefore they are forming these aggregates, soil aggregates. They are contributing to improve soil physical properties, but they also change the community, the functionality of the soil, the life that is in the soil. And this is very important. And this one, who has seen such a thing?
Tardigrade.
A tardigrade. So tardigrade is an animal that is known for being slow. So tardigrade is really means slow, an animal that is slow. So I think it is still quite cute, but it starts to be a bit different from what we are used to see. So this is not really the form of lives we are used to find. And this one you must know for sure. What is this? This is one of these animals, small animals that we associate with water, but we cannot forget that there is water in our soils. There is always a film of water, and these species that are characteristic of uh, uh, water ecosystems are also present in the soil, and they are very important in the soil food web. So at this point, I think I came to show to you a series of different forms of life that are present in the soil, in a living soil, in a good soil, and that have to share the same space, the same resources, and maybe they function in a very different way. But the idea is that this functionality is very complementary. And it is this complementarity that provides the characteristics and the functionalities of the soil. Is these different habitats, this heterogeneity that makes us healthy soil. And in this video, what we are watching is the difference between a soil that is not full of life, that is the one in the left, and comparing what happened when we have life in the soil. And so all these organic matter, all this litter that was in the top layer is being transformed, is being incorporated, is becoming alive, and we are producing very recalcitrant and stable organic matter. And this is really what we need if we want to sequester carbon in the soils. So from these top layers, if we go into a much smaller scale, we find acres, for instance, that all of us know about because of the allergies and other diseases, nematodes that are a nightmare for the farmers because farmers know some that are terrible and they think that all of the nematodes are bad, but not. You have to think that nematodes are the most abundant group of animals in the soil, and 4 out of 5 animals are nematodes. And they have incredible functions in the soil, namely regulating as regulators of bacterial and fungic community. But soil is not a safe place. And what I'm sharing here with you is a photo that took more than 1 year for us to have it. And what it shows is here in pink, an acre, and in blue, a nematode. And what we are looking at is a proof of the soil food web where the acre is eating the nematode. And this is relevant when we go to, we want to understand the ratio between animals and living organisms in the soil. These here are some amoebas. They are very important because they are kind of cleaning systems in the soil. They degrade virus, bacteria, fungi, etc., and they maintain soil salubrity. And in this slide, what we have is these green algae, another group that we associate very much with water systems, but that they are very important in the soils as they are primary producers, and in the absence of higher plants, they are the source of carbon for most of the living systems in the soil. And here, of course, we have one of the major components of the soil, fungi. Fungi are very relevant because they are important in aggregating the soil particles, and therefore they contribute very much to soil stability, erosion prevention, organic matter accumulation, etc. But they also create interact with plants, and they go inside the plants. You can see here an ether going inside the cell and ramificating inside the cell, producing this herbuscule. This is a mycorrhiza, endomycorrhiza, and this is an herbuscule. It is a magnification of the interaction between the fungi and the plant, and it's an area of active interaction between two organisms that belong to completely different kingdoms. So this is really an example of the communication, interaction, symbiosis among two individuals that belongs to different kingdoms. So it's what we call inter-kingdom communication. And this is really necessary for the well-being of the soil. Here is really a way of showing here the fungi. This is the root surface, and here we have the fungi penetrating the root. So is what we call the entry point. But apart from these fungi, we also have bacteria. Bacteria are very small, are always unicellular, but they do not live in a chaotic way in the soil. They are very much well organized at the surface of the different systems that live in the soil. And here we can see each one of these spheres is a bacteria, and then what we see here is exudates that creates a network that is a biofilm, and this biofilm really transforms the bacteria. A bacteria that is integrated in a biofilm behaves in a completely different way from those that are in having a free living style. And here in this picture, we can see the roots here. And here you can see many bacteria. And you can see here in this area a kind of membrane or gelatin or whatever, mucus, that is surrounding the root tip. So what I was asking before, how many of these root tips are present in a plant, it was to this structure that I was referring to. So this is the end of the root. We have here a meristem. So that is a part of the root that is giving cells dividing and allowing the root to grow. And my question is, if we have here a cell that is dividing and producing cells to the left and cells to the right, so after a certain time, this meristem should be in the middle of the root and not at the end. So why is it that this meristem is always at the end? Sorry, I hear that somebody said something, but I couldn't understand.
Sure, soil food web guy.
Because it's the cell dividing part of the root. It's where the cell keeps on dividing. No?
Uh, I didn't understand, but I presume that is quite close. Is that we have here a meristem that is dividing, so cells to the left, cells to the right. The cells that are produced to the left belong to the plant, so sorry, they will be part of the root, but those that are produced to the right, they are used to leave the root into the soil, the surroundings, into the rhizosphere. And it is in part these cells that are going to feed all these microbiome, all these microbiota that are in this rhizosphere. So it is the content of these cells that are going to be the food for these biota. So these biota have to interact with the root in order to make the root produce what they want, and the root has to produce certain compounds that select for the microbiota that the root wants to interact with. So you can see here that what we look at this picture and we see cows. We do not see any order in the bacteria that are around the root. There is a very complex communication in the way that these bacteria and the root interact to coordinate their activities and to have a good life. And this is a much nicer picture of a root tip, an apex, that is this white part, so that it is this conic shape, and that is populated on the top, on the surface, with bacteria that are creating a biofilm, so that are a form of close interaction with the root. So after having presented some of the main actors of the soil, what I would like you to understand is that they do not act alone. They live in this place that, as I said, is made by inorganic matter, air, and water. There is no light. The resources are very, very limited, and we have to have very regulated food webs that are allowing for the production of healthy soils. So we have been looking at acres, nematodes, amoebas, here the roots with the bacteria, with the fungi that are these filaments here in the soil. And what I like in this video is the way it provides a picture, an image for the interaction between these fungi, the bacteria, and the plant. So the plant is producing, so we are going through the mycelium network that surrounds the root, and this root really, it's not static. It's a complex structure that is interacting with the fungi, the bacteria, all the biota that are around, and it is influencing. It's providing food, but also information. Also, it is changing the way that all the other components of the soil are behaving. So having these dynamics in mind, and now I have to ask you if you have questions or if there is something that it's not so clear.
Is there any question?
Sorry?
Is there any question at the moment? No, I think it's very clear and clear.
Very clear. Okay, thank you. So this is the first part of what I wanted to show to you, is this diversity and the way that the diversity is working, is really functional. And now how we need this diversity and this heterogeneity to have a healthy soil, and how this is really a tool to sustainability and resilience. And this is not coming from the vacuum. For instance, all of us know that there are plants that can tolerate very high levels of stress, independently of we being talking about drought or high temperatures or whatever. And in this case, we are going to talk about plants that can stand 50.8 degrees centigrade. And they are still alive, they are still functioning, they are still green. And we were asking, how is this possible? And this took us to a fantastic discussion. That is, we all know that when we want to have plants that are well adapted to climate change, or stress, we try to breed different plants based on traits that we find in the plants, and we try to breed for those traits, or we try to find to modify plants genetically to have a genome that is providing a better phenotype, more adapted to the conditions we want to face. However, this has not been very successful, and we were asking why. Why is this not working? And one of the answers that we found is that most of our research and most of our work was based on looking at individuals. So we want, when we think about the theory of evolution, or the work of Mendel, or even the DNA and the work of Watson and Crick, we always think about the possibilities of an individual. But today we know that what we think that is an individual is not an individual, is an ecosystem, is a holobiont, is a microorganism full of microbes. And these microbes are everywhere and have a function. They're doing things. So all of us use the Google, but I think that not all of us know what a Google is. But a Google is a one followed by a big, huge number of zeros. And this is a very big number. And when we talk about microbes, we talk about Googles of living organisms that are present in all the surfaces. And they can change the way plants behave, animals behave, everything behave, because they are in symbiotic relationships with them. And when we went to look at this plant here that is really facing 50 degrees centigrade and still working, we found that in order to be alive and be active at this temperature, the plant has to be in symbiotic relationship with this endophytic fungal that is a curvularia. So it is these fungi that is providing the resources that the plant needs to be alive at these high temperatures. So our work nowadays is most of the time understanding what the plants are associating with when they need help. So when plants are under stress, we say that they cry for help. And when they cry for help, what does this mean? This means that they are producing certain exudates, certain compounds that are going to attract the microbes that they need. And nowadays we still have a lot to understand, but we have certain relationships that we understand. For instance, if a plant has iron deficiency, it it produces coumarins that are going to attract microbes that can chelate this iron. For instance, if a plant is in need of phosphate, it is going to formulate symbiotic relationships with fungi that are good phosphate scavengers, and they are going to produce these phosphate, to solubilize this phosphate. So this slide is quite complex, but what we need to understand here is that we know only a little bit of these dynamics, but at the moment we have some snapshots that allow us to relate the plant needs, the chemistry of this plant mediating this microbial recruitment. And what we think is that if we have a healthy soil, all these microbes are in the soil and the plant can recruit whatever they need. And we can test it through different tests, reflectometry, whatever, we can understand it. And all these corresponds also to different gene expression at the plant level. So this interaction of the microbes with the plant, we know today that involves a chemical communication, but also the plant with the microbes expresses a certain type of genes, and the microbe in presence of the plant also change the gene expression. And this is what is here represented. It is a plant that was not inoculated, and we are looking at the genes that were expressed in this plant, and two plants that were inoculated, and we see the difference. So the plant, the genome, the part of the genome of the non-inoculated plant is completely different at the one that is inoculated. So this difference in genome expression have to be related with the phenotype that can be drought resistance, heat resistance, etc., biotic resistance. This is a more complex slide that is showing that we know what are the genes that are affected, but we have to think that not all the plants in presence of the same bacteria activate the same set of genes, and not all the bacteria have the same effect in all the plants. So there is here a certain specificity that is very important And this is the reason why one biofertilizer, or whatever we call it, is not a solution for everything. Here in this video, that is a video we made as part of a cost action, I would like to
Biology
show
is
you.
extraordinary. It explains life. Pests and diseases cause losses of, but the spread of these scourges also. This is the progress.
Okay.
Several investigations have already shown bacillus, trichoderma, how mycorrhiza, and metarhizium are effective in helping the plants defend themselves against attackers, acting as bioprotectors or biopesticides. Let's take a look at a couple of clear examples that show how to apply what has been discovered so far. As mentioned before, bacteria and fungi that live inside the plant without causing any harm can modify the plant's chemistry, making them less suitable food for herbivores and improve their defenses. It has been proven that plants that have been bitten by caterpillars produce a volatile chemical to attract parasitic wasps, natural predators of caterpillars. When colonized by some of these beneficial microbes, the production of volatiles is enhanced, increasing the attraction of those natural enemies of the herbivore. These beneficial microbes are very common in plants, but so far they have not been thoroughly investigated for their role in plant defense. Not only can they help to increase resistance against insect pests and diseases, but also in improving the quality, nutritional value, and the taste of fruits, vegetables, and other crops. As in humans, we can vaccinate the plant so that it can later defend itself more effectively against herbivores. Bumblebees spend sufficient time visit microbes have many beneficial effects on plants besides protection is seen in the case with bumblebees and strawberries. For the cultivation of strawberries, it is very important that bumblebees spend sufficient time visiting the flowers. This is important to cover each of the pistils with pollen. If this does not happen, strawberries will become malformed and therefore discarded for sale on the market, with consequent loss of time and money for the producer. So it is very important to ensure and increase the amount of time that pollinating insects spend on the plant. Inoculation of plants with particular microbial root symbionts called mycorrhizae can increase the time that pollinators spend visiting the flowers, leading to more efficient pollination and increasing crop yields.
Okay, so this is what we can do, and then you can see the video, all the video, but just to show you two examples of the work using this type of knowledge. For instance, here we have a crop that was inoculated and not inoculated, and we can see the difference in their resistance to drought. So maize that is being produced with of the water needed usually for the producing the same amount of maize. And here is another work where we were working with vineyards, and we could explain the terroir of certain vineyards based on the microbiotic composition of the roots. So there were several species that were indicators, and if they were there, the terroir of certain wines was present, and we could characterize and differentiate the 4 types of wines produced in different types of vineyards based on this technique. This is important because this can be told as a story, as something that connects our product to the land, to the soil, to the place where it comes from. And it is because we know these and we want to know more, but we understand that it's not the knowledge is not only on our side. There are other types of knowledge, saberes in Portuguese, that are in other types of actors, that we think that citizen science is very important. It is, for us, it is very important to involve all the citizens in this type of discovery, and to profit from the contribution of everybody. And this is what ECHO is about. ECHO is a European project. It started almost 1 year ago. We have been developing the tools during this year. It has 16 partners, but it wants to cover all the European space, and we want to involve the 28 European member states. We want to develop a toolbox that is going to be a free resource to disseminate soil among the community. We are going to develop a set of 16,500 soil analysis, and we are going to assess the biology, the biotic community of these samples, and all of these is going to be an open source in a repository that is going to be part of the EUSO, the European Soil Observatory, but it's going to be available to any type of stakeholder. And so in order to have these activities and to better involve the community, we have a system to create the most possible impact. We have developed the concept of having soil ambassadors. And these soil ambassadors are a kind of, interplay, intermediaries between the people that are working in the soil in a certain region and us, and are giving us the questions, the worries, the problems, and also connecting us with their own land. And we are starting this process. It is a very important process for us, because we have been developing tools, for instance, that show how to choose a good spot to collect a soil sample, how to what are the rules to collect this soil sample, how to manage it, and what we have to do. We are going to start this collection on January 2025. And I would like to use this opportunity to invite Val Dalama to be one of our soil ambassadors, and to better connect us with the soil of the south of Algarve, that is so peculiar and and it's not well known, the limitations, the biota, the interactions. I think we still have a lot to understand about it. And after this, I just would like to invite you to go and visit our website, and to thank also you for your attention, and all the collaborators of our group that are many that produced and allowed us to have all these information that we could share today with you. So a final thank you for your attention, and please put your questions, your doubts, everything.